Okay, hi. So we are going to go through B2 now. So we'll do a page on B2.1, a page on B2.2, and a page on B2.3. So B2.1 probably has the most in it. So quite a bit of SL material and also some fairly challenging HL material. So we'll start there, but it does get a little bit lighter as we go, which is good. Just in terms of spacing, so here's going to be our one page. So I'm going to draw essentially a membrane in the first half, and then in the second half, I want to go through basically the 5 specific types of membrane proteins you need to know at an HL level. So top half is SL, bottom half is HL only. So if you're SL, take some space. You have the whole page. If you're HL, try to keep your membrane to half a page if you want to stay with the one-page summary idea. Okay, so we are going to draw a phospholipid bilayer. And what I want to show on the bilayer is all the different components and the processes. If you were having to draw it on an exam, you wouldn't have to actually include like the names of the processes that I'm putting down there as well. You could just draw the membrane, but I just want to kind of cover them all in one place because they are so linked. Okay, so there's no amazing rhyme or reason for how many phospholipids I'm putting between proteins, so feel free to kind of roll with what works for you. So we're going to draw our bilayer. I'm going to try to remember to draw some of my fatty acids as unsaturated. We'll label that kind of as we go. But again, it doesn't have to be any set number. I'm just trying to remember to show that. So I'll do that here, like so. Okay, then I want to draw proteins. I'm going to draw kind of 3 different proteins that represent the different processes. So I'm going to draw here a channel protein. So I'm going to draw a channel protein with kind of the open shape of it. So we're showing kind of a pore in the center. I might even go ahead and show that this is creating a polar core. So it's creating kind of an opening in the membrane with polar kind of flagging it. All right, then I'm going to draw a couple more phospholipids. Then I'm going to draw a very specific type of protein channel as an aquaporin. They have a lot of alpha helixes, so a lot of times you'll see them kind of drawn spirally a bit, just representing that, but it's not necessarily, you don't have to represent it that way. Okay, then I'm going to draw a few more phospholipids. I'm actually going to draw a couple more here because I want to add some peripheral proteins. So I'm going to give myself a little bit of surface to do that. I'm going to not forget to draw some unsaturated ones. Perfect. So on there, I do want to use this as a little place to show that some of my proteins are actually peripheral proteins, and that those can exist on both sides of the membrane. And then I'm going to draw a protein pump. So I'm not going to worry about drawing this completely accurately for the sodium-potassium pump. So there's nor am I drawing it particularly well. We'll do the sodium-potassium pump in the HL section. You just need to know at an SL level that there are protein pumps that are in charge of active transport, so you don't have to know that one specifically. Okay, and then what I want to draw in this last section, because I'm trying to include membrane and transport, is I actually want to show some endocytosis. So I'm actually kind of showing my membrane moving inwards, like so. And then, because I want to label it here, there you go, I'm going to draw an unsaturated fatty acid, like so. Okay, so there's my, sorry, these two are not very well drawn. I'm going to move them a little bit lower. Oh no, that's good, they can be higher up. Okay, so there's my membrane. If you were drawing it for an exam, I would probably stop it about there, so you don't need to show the endocytosis. I'm just doing that for this sake. And then let's kind of make sure we remember what we are drawing. So this model that shows the proteins throughout the phospholipids is called the fluid mosaic model. And again, the fluidity refers to the fact that the phospholipids are not bound to one another, allowing for movement, and the mosaic refers to the proteins kind of throughout. Okay, so let's start with the bilayer and kind of make sure we remember this. Came out quite a bit in kind of B1, some of the molecule stuff, so hopefully we can recall some of those details. So you can put it at whatever end you have more space. I'm gonna just put it at this end, but we just wanna label one. So remember that our heads are polar. And again, you can go back to kind of your B1 material and revise why that's the case. For this topic, you just need to know polar versus non-polar. And that's on both ends. And a reminder that that makes them hydrophilic. So these are hydrophilic, which is great because both the external and internal environment are aqueous. So having the hydrophilic heads at both sides allows it to be kind of surrounded by water. And then the tails are going to be our non-polar because they are made up of lipids, fatty acid tails. And they're going to play a really important role in maintaining structure of the membrane by having a component that would not dissolve in water. And so those are hydrophobic, meaning not attracted to water. And again, covered more in different areas, but remembering from kind of, I think, A1, the idea of spontaneous formation. So when you have A2, when you have vesicle like phospholipids, because of their properties, the hydrophobic tails will automatically turn towards each other and create a bilayer kind of spontaneously. So that's a really kind of good little remembering. And so again, that makes up the bilayer. Another little reminder is that because they have both polar and non-polar aspects, we can actually say that they are amphipathic, and that's a term we should have learned kind of in other areas, but it does show up specifically in this actual, apparently I can't spell amphipathic. There we go. It is named in the kind of syllabus for this section, so just a good idea to remind ourselves what that means. On that note, there's another amphipathic thing up here, so let's kind of clarify that. So this is an example of an integral protein because it's sitting throughout the membrane. And the only way that it can be throughout the membrane like that is because it is amphipathic as well. So it needs to have a non-polar regions kind of surrounding the phospholipids. So this whole area here would be like non-polar, non-polar, and then it would need to have kind of polar regions up at near the phosphate heads. So just a reminder that your integral proteins are also amphipathic, so that's a term we need two places. These little proteins here are my peripheral proteins. And I have drawn them both places. Just as a reminder, they can sit on either side, but they actually do not go through the whole membrane, and as a result, they do not have to be amphipathic. They are usually largely more polar proteins. They may have a non-polar core, but they can kind of be polar, kind of globular proteins, so they have a slightly different structure. And then one of the important things they do is kind of attach to the cytoskeleton, so I'm just showing that there. Okay, in terms of things that we're going to actually draw, so I'll do the transport types last. I want to get all the bits and pieces of my membrane. So while we're talking about fluidity, we need to kind of comment on two things that aid in the fluidity. So I drew the unsaturated fatty acids. The other thing that in animal cells is really beneficial is to have cholesterol. So remember, cholesterol are these kind of steroid fat, like steroid lipid chains, and they're not in every one, but kind of throughout you'll see them, and so they sit in between our phospholipids. I didn't leave a lot of room for them. Okay, so I'm going to make a note of that. It's kind of hard to show it when I'm showing endocytosis there, but just for spacing reasons. So cholesterol plays a really important role in animal cells. So we have cholesterol, and it actually works to kind of maintain fluidity at any sort of extreme temperatures. So at really cold temperatures, it provides a spacing, so it doesn't allow them to condense. At really warm temperatures, it actually provides, like, the tails are attracted to it because of its steroid lipid properties, and so that it kind of holds on to them. So it's not a bond, but it does kind of pull the tails inwards towards it. So it actually kind of works in both ways. So it actually preserves fluidity at both temperature extremes. So it's a really great adaptation because of its ability to modulate both kind of temperature extremes. So it works really well. Unsaturated fats, on the other hand, are very distinctively helpful for one purpose. So I might put that one here. So these are less helpful in warm temperatures, but they are actually going to maintain, like, prevent rigidity in cold temperatures. And so plants, all of their maintaining of flexibility is done this way. They don't have cholesterol. Remember, they do have cell walls, so they have a few less threats than in terms of warm temperatures. So because their overexpansion isn't going to burst the cell due to the cell wall, they don't have as much concern there. So they generally just have the adaptation for cooler temperatures. And so these prevent rigidity at cool temps. So those are going to prevent it from kind of getting too tight at the colder temperatures. So depending, and it's an important adaptation, so plants and animals, so we also have unsaturated fats. So organisms that do reside in colder environments have more unsaturated fats. Organisms that live in more moderate environments might have a balance. Organisms that live primarily at warm environments probably have more saturated fats. So it can work as an adaptation, which is really cool. So we have our two things that, again, are both fluidity related, so maintaining shape. The last thing we need to draw, I didn't leave as much room as I probably should have on top of my membrane. So we'll draw our glycoprotein. We could also have a glycolipid. So I'm just going to draw it kind of coming off of there just to show that you can have either of those two things. So those are going to be my glycoprotein and glycolipid. And they have a similar function. So they have kind of two things they are important for, which is essentially adhering. So they can be used as adhesion molecules. We'll talk specifically about CAMs later at an HL level, but they can act as a way of kind of the glycoproteins can link and create kind of an adhesive surface. Or in some animal cells, they have so many glycolipids and glycoproteins, they basically create what's called a glycolax, which is kind of a whole carbohydrate sugar layer, which can help the cell stick on. So they play a role in adhesion. But they also play a role in identification. So you don't need to know tons about them other than those two purposes. So adhesion and identification. So again, somehow my real one wasn't quite as crowded, but that's okay. So that's everything I need to put up there. So we're good now. So that's kind of, if you were drawing a phospholipid bilayer fluid mosaic model, you could basically, these could be upright, but those are the kind of features you would need to draw. So the different types of proteins, integral versus peripheral. You need to remember to draw your unsaturated fatty acids and your cholesterol. Label your parts of your phospholipid. What I'm adding to this one is I want to go ahead and draw the different types of transport on the same diagram because they are so related. So what I want to look at first is we're going to kind of build in complexity, so to speak. So the easiest way for things to move in and out of cells is with simple diffusion, and simple diffusion happens through the phospholipids. So we're going to show that here. So I have outside my cell quite a bit of oxygen, like in the bloodstream. Inside my cell is less oxygen. So what it's going to do is it's so small that it's going to fit straight through the tails. and then it's gonna move inside. And oxygen can do that really easily because it is non-polar. So the tails it doesn't try to move away from the tails. It can just move straight through them. So that facilitates that process. So, sorry, I shouldn't have used the word facilitated for simple diffusion. So that is simple diffusion. Simple diffusion, all types of diffusion are passive transport. So I'll just kind of pause and add what do we mean by passive transport. So when we say passive transport, we mean two really important things to know. One is that it means we're going from high concentration, that's my little concentration symbols, to low concentration. And another way you can say that is that you're going with the concentration gradient. And then because that is the natural way that particles move, passive meaning no energy. So no energy required. So if you are being really passive, then that means you're not expending any energy. So if you're just passively watching something, then you're not using any energy. So that's our kind of what is true of passive transport. So for simple diffusion is non-selective. So we use their selectively permeable. So it is non-selectively permeable, which just means that there's no receptors. So anything that's the right size can fit in. So non-selectively permeable to small and uncharged particles. So the small is really important. So if it's large, it's not going to actually be able to get in. But if it is the right size and uncharged, it can move in. There are implications for that in things like medications. So if medications can be made to be small and non-polar, they can actually enter by simple diffusion because simple diffusion does not rely on any sort of receptor. So it's not specialized as long as they meet those criteria. So small is really important. When we say uncharged, preferably we mean non-polar. So non-polar are going to move quickly. Polar things can move but slowly. So they generally don't move through simple diffusion. Just the reason being that because of how slow it is, we usually have adapted to have a facilitated option. But they could eventually make it through. They just get kind of stuck. They kind of are moving away from the tails. And so it just takes longer. It becomes a bit of an obstacle course for them. So generally when we're talking simple diffusion, we're talking small and non-polar. So things that are small and non-polar move quickly. I drew oxygen because it's a really important example. Oxygen and carbon dioxide move by simple diffusion, which is really important for gas exchange. And as we go into specialized cells, their ability to move by simple diffusion is really important. Okay, so then the next place along, I have a channel protein. So now I want to show something that is having to move by a channel protein. So this could be something that is charged. So let's say I have calcium ions. I need to try to draw a concentration gradient in my very limited space. And then they're going to move through here. So there's my movement. I'm still going from high to low. But this time, because of its charge, it would not be able to fit through the actual membrane. The charge would repel the phospholipids so much that they would push it away. It wouldn't be able to make it through because of its high charge. So it's gonna have to go through a protein channel. And as we showed, the protein channel has created basically a polar area, like a polar gap in the membrane, and it's gonna go straight through those. So this is also passive. So it is still not using any energy. So basically that pore is there and it's available. A lot of times this is selective, meaning most of the time particular channels only work on one ion. So it's usually selective. So it usually only acts on one or two kind of particles. So it's not often just an open channel for everybody. I don't know why I wrote that in the wrong color. I'm going crazy. Sorry, you can leave it, but I'm going to be anal and change that. So it's selective to specific particles. So sometimes one can actually let a couple ones in. A lot of times they are single particles, so they are actually bound to very specific things that they can let in. And then the specific particles, they can be slightly bigger, but the key here is that they are primarily polar or charged ions. You can sometimes get slightly larger, nonpolar. So if you had something that was nonpolar but not able to move through the phospholipids by size, it could be done with facilitated diffusion if there were an actual kind of channel specifically for that. The other thing is your course does not require anymore for you to know channel proteins versus carrier proteins. So I have very specifically drawn this as a channel protein. But do note that this is also done with carrier proteins that actually have like a shape change. So when the thing binds to it, they change shape. So we're going to focus on channel proteins because that's all the examples, so you don't have to actually know any carrier ones. But that is another option for something that can also be used for facilitated diffusion. I'll make a note of that just in case you're looking at kind of old course questions. You did have to know that. So also carrier, sorry, maybe that wasn't very clear, carrier proteins. Okay. Then I have a specific type of facilitated diffusion. So you'll notice this one here is intended to be an aquaporin. So an aquaporin is a protein channel. So it's the same concept as that one. It's just specifically, and we said they're usually specific. So all protein channels have kind of specific ones. This one is specific to water. So if you remember, you should remember this because it came up with what we're talking about there. Water is polar. So it's not charged. So in theory, water can move via the membrane, but we move water so often that we really want the process to be efficient. So osmosis is a really, really vital process. So by having aquaporins, we can make it much more quick. And cells that do a lot of movement of water often are very rich. I'm writing the word aquaporins again. They're often very, very rich in aquaporins. So we'll come to that when we do the kidneys, but basically looking at kind of how we can vary the number of aquaporins to increase water movement. Osmosis is passive. It is actually a form of facilitated when it goes through the aquaporins. So it's facilitated diffusion by aquaporins. So facilitate diffusion of water by aquaporin channels. And remember that it could happen slowly by simple diffusion. So it is possible for it to happen just by simple diffusion, but most of the time we're going to use the aquaporin channels because it's a much faster process. So all three of these have in common that these are all passive transport processes. So all three of them move by passive transport, and so they are able, and I'll write the word because I didn't use it before, passive transport. So all three of these are moving with the concentration gradient from high to low concentration and not needing any ATP. The next two processes that we're going to draw are active transport. So that's why we're kind of showing that as a distinction. So here I have my protein pump. So protein pumps are one of the big ways the active transport occurs. And so with a protein pump, the key is we are now moving it away from its concentration gradient. So let's say inside here I already have a lot of sodium. But I still want to move sodium in. So I'm going against the concentration gradient in doing that. We're going to do the sodium potassium pump at an HL level, at an SL level. It's a great example, but you don't have to know specifically how it works. So this is going to be active transport now. And we're going to show also endocytosis as another type of active transport. So active transport is active. What do we mean by active? Active transport means we're actually this time going from a low to a high concentration gradient. So we're actually moving against the concentration gradient. So this time we're going from low to high. And we can also phrase that as against the concentration gradient. And then because of the fact that we are going against the concentration gradient, we use ATP. I'm going to write ATP or energy. There are some that work with using like excited electrons, so it's not always ATP. ATP is a very common one, but there are kind of other forms of energy that can do that. So active transport, so the example would be like the sodium-potassium pump. And the key is it can move anything as long as it's against the concentration gradient. So it can move ions, it can move nonpolar, polar particles. The key that makes it active is that it's against the concentration gradient. So while these are kind of more specific in terms of what type of particles, active transport can be involved in the movement of anything. So for instance, sodium can be moved by facilitated diffusion or by active transport, just depending on whether we're moving it with or away from the concentration gradient. Okay, and then the other thing I added here, just because I wanted to kind of include and show that there is another form of active transport, is going to be endocytosis. So at an SL level, you only have to know kind of by name. At an HL level, you have to know a little bit more. So I will kind of cover the slightly bit of extra HL information, but it won't kill you at an SL level because you do have to know what they are. So, which also reminds me that like SL, you don't actually have to know about unsaturated fatty acids. So that's a HL only thing, but it's not a bad thing to know. So I didn't worry about putting it on here. Okay, so what we are showing here is endocytosis. And endocytosis is an active transport process, so it does actually require ATP. And generally speaking, the reason for endocytosis is often to move a larger particle than you can move with the actual protein channels. So it's also bringing in larger particles, but it can be done with like dissolved particles, so you can actually bring water in. But most of the examples you need to look at are involving kind of the larger particles. And when they come in, they actually push through the membrane and they end up in a vesicle. And we'll actually show an example in the next section. So we'll look a little bit at phagocytosis of pathogens as our example. So you do need an example, but we'll cover it there. So that's endocytosis. And then exocytosis, on the other hand, also going to be active. So we're still going to use ATP. Just making sure you can see that barely. The difference here is that we are going to remove larger particles. So a good example of that is proteins, which again we'll pick up later. So we're going to remove larger particles, and they are in vesicles already. And the vesicle merges with the membrane. So the way, sorry, I'm gonna have to move that slightly, I think. Sorry about that. So the way that works is, again, it's already in a membrane from the Golgi, it comes to the cell membrane, and then the actual vesicle merges with the membrane and it comes out. So that's a little bit of endo and exocytosis, which again, at HL, you do need to be able to account for kind of what's going on with the vesicle. So the idea of formation of a vesicle versus merging of a vesicle for the two different processes. At an SL level, you just have to know basically in versus out is all that's required there. Okay, so that's all things fluid mosaic model. So that's all things SL with a couple of bonus material. The next section is HL only. So if you are SL, fast forward for a bit and go to B2.2. But for HL, there are essentially 5 different specific integral proteins that you need to know about. So we're going to kind of cover those ones that are kind of by name that you have to know. So this is kind of general, but there are some of them that you have to know by name. And we're going to try to look at kind of what do you actually have to know right now. A lot of these are proteins that are going to come up when we get to physiology. So when we go through organisms and we get to kind of physiological processes, these are proteins that are going to be important there. So right now we just need to understand how the protein works. We don't need to understand the whole process. So I will link them to a process just so you have that kind of idea of where it fits in, so that if you're watching these to revise, you kind of make those links. But for this part, it's really just how does the protein itself function. Okay, so we're going to make a table for this. So these are going to be specific membrane proteins to know. And I am going to indicate, again, just so maybe write this in your notes as well, that this is HL only material. Okay, so what we're going to do is we're going to have a column, pretty simple chart, column where we name the protein. Then we're going to do a drawing of the protein, including in some cases if it goes through kind of changes. So leave a bit of space for your drawing column because I do need to sometimes show kind of things happening. And then we'll put functional info, which means kind of what is it related to for its function. So a little bit more information for that. So the first 3 are all related to neural transmission. So once we do neural transmission, you're going to see kind of where this fits into a context. So it's a little bit strange being out of context here. But we need to essentially know about these proteins in terms of what makes them open and close, how they work. Again, not necessarily the content around their functioning. So first up, we have which is basically a neurotransmitter gated ion channel. So a neurotransmitter gated ion channel. And there is a specific one that they want you to know. So it's nice sometimes when they name it. So the specific one they want you to know is the one that is done with nicotinic acetylcholine receptors. Okay, so that is the receptor. So the actual membrane channel, so it's an ion channel that has a nicotinic acetylcholine receptor. Acetylcholine is the neurotransmitter. These actually respond to nicotine as well, which is really interesting. So that's one way that nicotine influences our body, is it fits into this acetylcholine receptor. So that's where the weird term comes from. But let's have a look. That is what they are officially called. So nicotine is just a substance has been created to act on these receptors as a way of kind of creating their impact. So this is going to be occurring in between neurons. So this is related to kind of a neuron releasing a neurotransmitter and then a cell that acts on that receiving it. So here we would have kind of our neuron. And this would be my next neuron or effector cell. A lot of times it's a motor neuron, but in some cases these can be directly on the muscle tissue themselves. So it can actually be released onto the tissue. So that's why I just wrote kind of effector, so the cell that it's acting on. And so this cell has an actual protein ion channel. Usually it's actually a sodium channel. So this is a channel that is intended for a sodium intake, but it is usually closed. So it is normally closed or gated. But what actually opens the channel is it opens in response to neurotransmitter release. So then this cell has neurotransmitters. This neuron has neurotransmitters in it. Acetylcholine is the actual neurotransmitter. So these get released. And then when it is received, then it opens. So I'm gonna cheat and draw that with my whiteboard marker, which is hard for you to draw. But basically it opens in response, and then when it opens, then my sodium can move in. So if it is open, then my sodium moves in. So sodium only moves in when the acetylcholine has binded to it. So that is the acetylcholine receptor. So this is the acetylcholine, which is a neurotransmitter. And in the presence of the acetylcholine, the channel opens. Again, this is all part of a complex kind of neural messaging, but for right now, what you want to know is that it's a protein. It is shut when there is not any message binding to the receptor. When the message binds to the receptor, it opens, allows the sodium in. These actually play a role in initiating action potentials. So in terms of what is our information, so it is gated, which means normally closed, which means the sodium stays out. So my sodium is not able to get in. They open in response to a chemical messenger. And in particular, the chemical messenger in this context is the neurotransmitter acetylcholine. So that is my chemical messenger. So I'm just going to star that and show that that is that. And of course, these play a role in neural signaling. And they actually are going to play a role in kind of initiating action potentials. So when the sodium starts to enter, that actually is what will spark the action potential. So don't worry about that right now, but when you're revising this and you actually know about neural signaling, make that connection that this is kind of the start of that neural signaling process in that second neuron. Or in some cases, it stimulates the action of the cell. So it can start a neurotransmitter or it can lead directly to action if it's kind of at the end of a neural pathway. Okay, so closely related. So I'm going to try to do all three of my neuron ones together, and then I'll have to erase. So the next option is also a gated ion channel, but it is a voltage gated ion channel. And the specific one that you need to know is potassium. So our specific example is the potassium channel. And this plays kind of the next role along in the action potential. So we are actually going through kind of three protein pumps that play a role in action potential without covering action potential, which is weird, I admit. Okay, so what's happening here is here's my cell, and then here is my potassium channel, and it is normally closed. And you can kind of see how I've drawn it so that it's literally staying kind of shut. That's just membrane. And this happens when we say voltage. This is related to charge. So it is closed as long as the inside of the cell is negative. And that is the case until the sodium comes in. So that's where they're related to one another. And then once the sodium comes in, it actually introduces a positive charge. And this ion channel responds to the charge. So it doesn't respond to a neurotransmitter, it responds to a charge change. Oh, and just for accuracy, I'm going to draw this little ball and chain there. So now it is going to actually open up in response to that increase in charge. So that is going to now open. And what has triggered its opening is it going from negative to positive. So this is what has caused that. And then now that it's open, my potassium is going to be able to move through. So now I can move potassium in or out, whichever way. So now my potassium is able to move. It actually moves out. So my potassium here was locked, but now it's able to move. Then I actually want a short burst, and I don't actually want it to diffuse back through. So what will happen then is until the sodium potassium pump kind of returns things to their position, this actually has a pretty cool mechanism in place that actually swings around and locks it in. So then once the potassium has moved, that actually is going to keep it out. Otherwise, it would start to diffuse back in. So this is really helpful in the event that it is still positive right now. That's going to actually kind of keep it out. Most likely, this has a similar thing in place. We just don't actually know the mechanism. We actually know the mechanism for potassium is related to that ball and chain. I'm not sure exactly what the mechanism is, but probably a similar concept where it has some way of reshutting quickly so that the direction is one directional. Okay, so this is gated as well. It is closed when the neuron cytoplasm, just so that's the inside of the cell, is negatively charged. So that is kind of the normal state at resting potential. I use the word relatively. That's going to be important when we get to neurotransmission. It is negative relative to the outside. So anyway, it does actually have positive ions in it. That's fine. Just ignore me for right now. I just don't like to write things that are inaccurate. It's what we're going to learn later. But the emphasis right now is that there is a negative charge. They then open in response to a positive charge. So the axon, which is just the inside of it, becoming positive. And then they do also have that kind of quick shut mechanism. So that's what the ball and chain does. That's actually probably a little bit less important, to be honest, than just understanding that the open and close relates to charge. And that these are going to play an important role in continuing or propagating, fancy word, continuing the action potential. So essentially, this is going to play a key role. After the sodium is entered, the next thing that's going to happen is the potassium goes out, and that's going to kind of trigger the next response. So interesting, kind of more to come on that down the road. So both are playing a role in action potentials. Once you reach the end of the action potential, you actually have to kind of return everything to where it's supposed to be. And so that is actually done with the sodium potassium pump. Sodium potassium pump has several different important processes. So I'm drawing it here in a neural context, but it also plays a role in movement in intestines and kidneys, and a lot of times it will actually move the sodium in versus out. So it has different roles depending on where it is. So it's a little bit uniquely, it moves sodium and potassium all the time, but it can have kind of different roles. So the sodium potassium pump, so I'm gonna draw this as simple as possible. I think I'm gonna draw it in 4 configurations instead of 5. Normally, so I'm not going to draw the actual shapes like I did in class just to try to make this more of a summary. So normally it is facing open, and so what happens when it is faced that way is that I'm going to have 3 sodiums come in and bind. Okay? At the same time, I'm going to actually have a ATP come in as well. So there's my ATP is going to bind, and my 3 sodiums are going to fit. What's going to then happen is we're going to actually have ATP hydrolysis. So we're actually going to break that off. And what that is going to do is it's actually going to close my pump and spin it. So this is actually going to have just a little phosphate attached. So that's where the ATP gets used. And then my sodium is going to go out. So in one direction, so I made my shoes do all these hand motions, so it looks like this. When the sodium comes in and the ATP goes, it goes like this. So the ATP, sodium comes in, ATP opens it. Once it opens, the sodium leaves. And then I did the analogy in class where I basically said like, okay, this is the Uber driver that dropped your brother off and then you want to go out. So you're like, hey, I want to pop in and use that. So then that's essentially what happens. So then my potassium is going to go in. And then when the potassium goes in, that actually pops off this phosphate. So potassium goes in. So we were like this, now we went like this. Now potassium comes in. When potassium binds, it knocks it off. So then that is going to then change it back to this shape here. And so now the potassium is going to leave like so. And then we can restart the process. So then once it's like that and the potassium has left, then we can start the next thing over. So at the end, we're just restarting the process. Okay, so we said sodium binds, ATP binds to change the shape. Once it changes the shape, my sodium exits and my potassium enters. When my potassium enters, it knocks off the phosphate. So notice the phosphate is no longer attached. That actually causes the change back to the original shape, which means the potassium will leave, and then we will start the whole process again. So I think I did it in 5, but you could leave that off because it's just restarting the whole process. So there is my sodium potassium pump. It's also like moving downwards, but that's okay. Okay, so first, what do we need to know? So this is an exchange transporter. So it means for every transport we get 3 sodium moved and 2 potassium moved, basically for every cycle. So it moves the ions together. So we only have to use 1 ATP to move both. So essentially for every 1 ATP, we're able to actually move both of the molecules. ATP is actually used to cause the shape change. And it has 2 conformations. So that's kind of what we're trying to show here. It's either kind of open to the inside of the cell or open to the outside of the cell, and ATP is what does that. So ATP is what opens it, and then when the phosphate leaves, it's what closes it. So that's kind of the role that ATP is plays. And then these are actually used heavily in neurotransmission. They actually return it to resting. But they're also used with co-transporters, but I'm going to do those next, so I'm not going to put that there. So they basically return your neuron back to resting potential at the end. Okay, so again, you don't necessarily right now need to understand these parts, but you do need to know kind of how the pump itself works, which hopefully will be nice because then when we pick up the neural transmission part, you already basically understand the pumps involved. And so that's kind of one less piece then that we already kind of have covered. So then it's easier to kind of add in the theory. That's the hope anyway. We'll see. Okay, 2 more to know. Last one's easy. This next one's still a little bit complicated, in part because it's part of a relatively complex process. And so we kind of need it'll make more sense once we do it in kind of context, in terms of where it's kind of happening in the body. But this is going to be the sodium dependent glucose cotransporter. So sodium dependent glucose cotransporter. So glucose can move by facilitated diffusion if it's going with its concentration gradient. It is polar, so it is a challenge to move even then because it's size and polarity, so it requires a protein channel. But a lot of times we're actually trying to move glucose away from its concentration gradient. It's a really important molecule that we want to keep like in our bloodstream or go into cells. So a lot of times it has a very specific place it needs to be functionally that may not align with its concentration gradient. So this is a kind of tool that allows us to move it against its concentration gradient. And it actually makes use of kind of an existing pump that we already have. So rather than needing another pump, it makes use of kind of two proteins. So what we have here, so I'm going to draw them. I think I tried to draw them on kind of two sides of a cell, but I'm just going to try to draw them kind of along a cell to show this. So we have our sodium potassium pump. Okay. And then down the road from that, we're going to have a new pump, which is going to be this cotransporter channel protein. So this should be a channel protein. Just two, I think. Okay, so that is my channel protein. And like all we were saying before, a lot of these are somewhat gated or specific. So we can kind of consider this one to be a little bit gated. So it's going to normally be closed. It actually needs kind of energized sodium to open it. So when sodium needs to move through it with its concentration gradient, the energy of the ions having been transported against the concentration gradient opens it. A little bit more complicated in terms of where the energy comes from for it, but we're going to try to actually go through it, to be honest, on the slightly simpler side, because you only really need to understand it on the simpler end, and sometimes you can overcomplicate things. So this is my sodium potassium pump. So we are familiar with the fact that my sodium potassium pump can move my sodium out or move it in one direction. So it is actually doing that. And of course, remember, it's doing that using the energy from ATP. So what we're going to do is we're going to move sodium kind of against its concentration gradient until we actually build up a large gradient of sodium. So this is going to be a high sodium concentration. Once you have a high sodium concentration, that is going to basically open this. Go, and then it's going to allow my sodiums to enter, and then they are going to actually move through kind of by facilitated diffusion. But when they open up that carrier protein, it actually has a spot for glucose as well. So here's some glucose floating around as well in the outside area. And then the glucose is able to move in. So it moves at the same time. And it's actually moving against its concentration gradient. So again, kind of if you were going back to an analogy like an Uber ride, so it's already been paid for basically. So sodium has already paid for the move by using their energy, the energy to move the sodium. Sodium's already there, it's already paid for its ride. So it's already cashed its ride in on the app. It's already paid for, but hey, there's 2 extra seats in their lift. And so glucose pops in. So they're getting a free ride, but it's been paid for by someone else. So who paid for it? The sodium potassium pump used ATP to get this happening, Once that's happening, the actual channel responds to the high sodium intake, and it actually moves glucose at the same time. Actually quite similar to how ATP synthase works, where it moves from a hydrogen gradient that's been built up, is that's what kind of opens it up. It doesn't move something else, and instead it generates ATP at the same time. So that kind of is a repeated concept that sometimes we can activate a pump by movement of one thing for a second purpose. Okay, so that's kind of our functional info. So this is an example of indirect or secondary active transport. And that's because active transport is used in part 1 but not part 2. So we use energy to move the sodium, but no energy to move the glucose. So that's kind of where that comes from. So we use ATP to build the sodium concentration, but not, again, to move the glucose. So it's an ATP used kind of at one time only. And then in terms of where is this used, so it's actually used to move glucose against its concentration gradient. And it's done 2 places, so in the intestines and then also in the kidneys. So in the intestines, we're actually trying to move glucose out of the intestines all into the bloodstream. So we actually don't want it sitting, we don't need it in our intestines. So it's involved in kind of the mass movement of glucose out into the bloodstream. And then in our kidneys, it's involved in reabsorption. So that's kind of where it plays a role there. Okay, and then the last one we need to know is cell adhesion molecules. And this is going to be pretty straightforward. So these are CAMs. I kind of cheated there, but trying to use that. Okay, so you do need to know what that stands for, but also know that they're often called CAMs. And these are similar to what I was saying glycoproteins can do, but these can actually, that's not how I wanted to draw that, I'm so sorry. So here's my membrane of cell 1 and cell 2. And so these are not glycoproteins, they're just proteins. So then they are going to be integral proteins. Look like this. And then they create an adhesion point. So these play a role in creating a junction between 2 cells. So they create junctions between cells. And this is oftentimes really important for tissues. So a lot of times this is used to connect cells into tissues. And they can also be important because they can reduce kind of movement. And that actually has roles in kind of cancer prevention to keep cells from kind of removing away from its tissue that it's supposed to be in. So they can actually play that important role there. Okay, so that's all of this. Like I said, the next 2 will be a little bit shorter, but there's a lot to cover, particularly because of those hard HL proteins. So that's everything membranes. Now we're going to go into organelles and compartmentalization, which has a lot of overlap because it's a lot about membranes. Okay, so cell specialization. So kind of a weird topic. All thrown together. So a little bit on, it's basically on some special types of cells, which include stem cells, and then also some special cells within multicellular organisms in specialized tissue with a bit of cell size in there. So a little bit all over the place. So let's start with kind of the stem cell stuff and look at where do they fit in, where do they exist, and so forth. So I want to do this by kind of mapping the stages that are involved in kind of infant formation, because that's where stem cells play a role. I am going to put a few things above this chart, so don't write it right at the top of your page. Leave a little bit of space. So we make a new organism through gametes. So we haven't done a lot on gametes yet because we haven't done meiosis where I'm currently kind of teaching this. So just a reminder that gametes are haploid cells, which means they have half the DNA. So that's a cell that has only half the DNA. Okay, and then gametes, I don't like the angle this is at. There we go. And then gametes come together to form a zygote. And that process by which they come together is fertilization, which again is not something we've really gone into a lot yet. But that's the name of the process that the gametes form during. Then fairly quickly, that zygote then develops within about 3 days, 3 to 4 or 5 days, into an embryo. That embryo then develops for a matter of weeks. And then eventually, once it kind of has a fair amount of specialized tissue, we start referring to it as a fetus, and then an infant, of course, and then an adult. I'm putting all of those together because in the context of what we're looking at today, specialization is basically already where we're at. Most of it has occurred by the time it's a fetus. It already has kind of distinct organ systems. And then from that point onwards, it's kind of all the same. Okay, so let's look a little bit at these 3 stages in terms of what we can see for stem cells. So a stem cell is a cell that can self-renew and can make other types of cells. So it is a cell that has that ability. So at the zygote stage, kind of from the time that the 2 DNAs fuse together through to the first kind of 3 days of development, we have what are called totipotent stem cells. And you can think toti means total, right? So toti is total ability. So these are stem cells that have 2 important qualities. First of all, they can self-renew up to a point, so then they're going to generally turn into totipotent stem cells, and then they self-renew there, and they can basically form any type of cell. So they can differentiate into any cell type. Including things like placenta, umbilical cord, and so forth. That means they can make a whole organism. Within a very short amount of time, a matter of days, they start differentiating even the stem cells into kind of less flexible, more determined stem cells. So those totipotent stem cells, by the time we're looking at a 5 or so day embryo, are now pluripotent stem cells. And these are a little bit hard to remember, pluri versus multi, because plural means many and multi means many. So the toti is nice because it has a word built in there. But pluripotent stem cells also self-renew. That's always going to be a requirement for it to be a stem cell. And then they can become most cell types. So they can differentiate into most cell types, but not all. Primarily, it's already differentiated into kind of the outer layer that's going to become placenta and umbilical cord, and so it can no longer become that. So we kind of have 2 layers by that point, so it has some limitations. So most, but not all, and again, that's at a fairly early age. And then quickly from there, it starts to divide. So it starts to become, skin versus muscle versus inner organ tissue. And then from there, they become more specialized. So by the time you get to kind of an advanced fetus, an adult, infant, all of those, we have only left multipotent stem cells. So these are now also able to self-renew, but they have the ability to become many or few similar types of cells. So they can differentiate into different but similar, we'll say several, into several similar types of cells. So most of the time they're fairly limited, but a true unipotent or normal body cell can only become itself. So it's already a specialized cell and it can only become more of that exact cell type. So this is still a unique ability to go from one cell to self-renew and also differentiate into multiple different types. Okay, so a couple more things we need to add here. I'm gonna keep going on this for right now and then we'll add in a couple things. But where do we see these in adults? So we do still see multipotent stem cells, and they're found in what are called stem cell niches, which is just an area that still contains these. So again, they're not spread throughout, they're kind of localized. And you're supposed to know 2 examples, but it doesn't name the examples, so that means they can't really ask you specific details on them. So the main example that we looked at in class is the hemopoietic stem cells in the bone marrow. And those are interesting. So again, when we look at that definition of similar cell types, they have the ability to become platelets, red blood cells, white blood cells. So that shows that idea of kind of different types, but they're all blood cells, so somewhat similar in type. And then the other example we talked about that's in the book is hair follicles. We didn't go into a lot of detail about it because you would basically just need to list a second type. If you have to explain one, then you can explain a little bit more about the bone marrow becoming blood cells and so forth. Okay, so up here I want to go into a couple more things. So this, I've used this word differentiate. So this process here that is going between the different types is differentiation. And another way you can phrase it is cell specialization. So, but again, we'll use the same term. So sometimes you'll see it written as specialization. It's just differentiating into different types of cells. And anytime you see the idea of cell differentiation, we should automatically be thinking that that occurs by gene expression. So the way in which cells become differentiated is by turning some genes on and some off. So the turning on of specific genes. And so then the question becomes, how do they know what specific genes they're supposed to turn on? So the information regarding how they know this is basically coming from a signaling molecule. So they need some sort of external information that says, hey, you need to start becoming a heart cell. So they get some sort of prompting. There's one specific example that's really important in this early stage. And so this kind of early differentiation is done by something called morphogens. So morphogens are a cell signaling molecule, and they basically build up gradients. So the more morphogens there are, that tells it which type of cells it's going to become. So by kind of concentration. So it releases the morphogens early on, and then those morphogens settle into different areas and by different concentrations, and based on how many morphogens are in the area, that determines kind of what broad tissue type it's going to develop into. I don't see you needing to know in a lot more detail than that, but you could kind of just need to make that kind of example of what is a cell signaling molecule that plays a role in differentiation. Okay, so next question I want to look at that's related to this is, why do organisms grow by making more cells? So when this whole thing is happening, what we're noticing is that we're constantly creating more cells. I'm going to actually erase because this is a slightly different point, and this is still SL right now. I'm trying to form a link that's not really overly strongly there, like from stem cells to cell size. So my attempt at the link is kind of, why do we grow in the multi-celled organisms by making many small cells? In other words, why don't we just develop really big cells? So the answer for that is that you need to know kind of what large cells are not good at doing. So large cells have problems because they're too far away from the membrane. So there's too many components of the cell that are not close to the membrane anymore. And so they really struggle with diffusion. So large cells end up having a small surface area to volume ratio. So you do need to understand that concept. So they have a small surface area to volume ratio. You don't have to write the ratio, that is implied, which basically means they have a small amount of membrane. So they have limited membrane for lots of cytoplasm. So that can have its helpful things. So if you need lots of cytoplasm, that can help for like storage. But where it's problematic is they have generally really slow rates of diffusion. So because of the limited membrane, they have kind of very slow diffusion, which means they have a hard time getting things that they need or getting rid of waste. So limited membrane, slow diffusion of needed materials, which is problematic. So generally this is not as kind of beneficial. So this can be kind of negative. So I'm gonna draw a sad face. So the better option is to not have that. So instead, we wanna have small cells. So if we have small cells, they actually have lots of membrane per cytoplasm. So I'm just gonna again show that. So there's my cell membrane. So that is my surface area. So my surface area is the membrane. And then inside, the volume is the cytoplasm. Okay, so that's kind of what we're looking at. So these small cells, what's really important is that they have a high surface area to volume ratio, which is good. So it means lots of membrane, which means fast diffusion. So generally speaking, these cells are better because they have better access to the membrane. So that's a good thing. If you were asked to calculate that, so let's say you were asked to how you had to actually calculate that in some way, you should be given the formulas for that. So it is not listed in your dot points that you need to memorize the formulas. So you would just basically need to plug in the formulas. So if you were to have to calculate it, then you would basically go formula for surface area, and then you would go formula for volume, and then you would just divide them by surface area by volume. And so then your third step would be just to divide it. The more likely thing is that you just need to be able to understand and interpret. So you understand that small cells have a higher surface area, and that's a good thing, and that large cells have a smaller surface area, and that's not as ideal. But if you were asked to calculate it, it's a pretty easy calculation, but particularly for spheres, the formulas are a bit complex with pi in them. So don't worry about memorizing that, but you would need to be able to work it out if you were given it. Okay, so that's it for standard level for this part. So the rest is higher level, and the higher level stuff now is on specialized cells. So we're going from kind of general, like talking about the stem cells, to now looking at some specific specialized cells. And I think they've basically picked some examples that are going to come up again later. So I think it's kind of a handful of cells that we're going to go into the physiology of the tissue down the road. So it's worth knowing the format of the cells. So I think there's a rhyme or reason behind why they picked these random ones. but there are actual ones that you need to know. So you don't just need to kind of learn at your choice. It has itemized it pretty clearly in the syllabus, which is nice, that we can tell exactly which ones you need to know. Okay, so I've tried to do it by sum rather than just a giant table. I've kind of broken it up into 3 tables by kind of theme. So the 3 kind of groups of specialized cells they seem to want you to know is this group that are specifically specialized for a higher surface area to volume ratio. So some cells that are involved heavily in kind of diffusion, and so they have some features that are related to that. Then they look at kind of 2 different types of muscles, so looking at the idea of specializing within a tissue type. And then we're going to pick up a little bit more with gametes and the very specific specializing of egg and sperm cells. So that's our 3 categories. So we're going to label this whole section specialized cells to know. And then a reminder that this is an HL only section. Okay, so the first kind of group or category that we need to know, so we'll say category 1 is that our cells that are specialized for a high surface area to volume ratio. So what we were looking at previously is the idea that small cells have a higher surface area volume ratio, which is true. But there are ways to maintain a similar kind of size cell. You can't get too small because they still need to be able to fit in like mitochondria and organelles and nucleus and all that. So how do you kind of take a cell that's the right size for the rest of the functions but just increase the surface area? So there's 4 total types of cells that we're going to know for this one. So we're going to look at red blood cells, and we've kind of done these a lot. I'm just going to add the term erythrocytes. That is just a fancy term for red blood cells that they enjoy occasionally using just to make sure they can trick you up. The next type is going to be kind of related to the nephron, which we talked about a little bit previously in terms of the sodium-potassium cotransporter, or not sodium-glucose cotransporter. So this is similar kind of relationship to that. So these are the proximal convoluted tubule cells in nephrons, which are kidneys. And again, you're going to do an HL, we're going to do quite a bit on the kidney. So again, I think that's just picking up something you're going to need down the road. And then the next 2 I'm going to kind of include together because I'm going to draw it one place. But there's 2 different types of cells in alveoli. So this is going to be type 1 pneumocytes and type 2 pneumocytes. Okay, so I'm going to try to stick to what you really need to know for this subtopic, because again, there's going to be more down the road, but kind of what is their structure and then why is that important. So red blood cells, again, we should have already kind of covered this pretty well. If you look at it like this, it would be about 6 to 8 micrometers, but in reality, it looks like this. And it has a diameter of about 1 micrometer, something like that. So it's biconcave is the shape. And then that's really helpful because I have lots and lots of hemoglobin inside, and I want my hemoglobin as close to the surface as possible. So that's why that's helpful. There's my hemoglobin. So biconcave shape is flattened. So we can call it a flattened biconcave shape. And again, I've already said above, all of these, the benefit is the high surface area to volume ratio. And why is that important? So it's easier for oxygen to load and unload onto the hemoglobin. So it has to basically by diffusion. So it needs to diffuse through the membrane and then load onto the actual hemoglobin, and then when it gets to where it needs to go, it has to unload. So oxygen has to kind of diffuse both places. So we want to basically have our hemoglobin close to the surface because it's much easier to pop in and out than if it's way inside the cell. It wouldn't be able to transport as well. Okay, next up we have these proximal tubular cells. So these are going to be a little bit different. So they're not going to be quite as small. So they're still going to have this kind of almost like square shape. And that's important because they actually need to have a lot of ATP made. So they have lots of mitochondria. So we can't have a tiny, tiny cell when you need a lot of mitochondria. So they would have like a nucleus, and then they would have lots of mitochondria all throughout them. And so you need to have a larger cell. So there's two things that are being shown on here that are particularly useful for increasing the surface area to volume ratio. So we have up here microvilli, which are those like little finger-like projections of the membrane. And then we have down here just the kind of less dramatic, but kind of fold-ins. Just call it invagination, which is just basically like a little fold-in of the membrane that also creates more membrane. So both of these are going to have more membrane, and that's going to allow for faster reabsorption in the kidneys. And faster reabsorption is, of course, done through diffusion. So all we need to know about these for right now is just those two kind of adaptations that they have that enable that process to happen more quickly. Okay, then over here we're going to have I'm going to draw one alveolus, which is one kind of air bubble in the lungs. And the majority of the surface is these type 1 cells that are super, super thin. Actually so thin that they like have a slight little bulge where the nucleus is, because they're so thin that even the nucleus couldn't fit otherwise. But then there's also these quite a bit larger type 2 cells. So there's our type 1 and type 2. So these ones here are going to be the type 1, and this one is going to be the type 2. Okay, so let's look at their adaptations. So type 1 is very thin, and the reason for that is it's very thin and it's close to capillaries that are also very thin. And they're going to also have kind of a 1-cell thick layer. And the reason why that is important is because, again, diffusion, so it's going to be faster exchange of gases by diffusion. And we talked about the idea in our first part of this that oxygen and carbon dioxide can both move by diffusion, so that's a really important process. So then the question is, what is going on here with our type 2? So these are larger cells, so they're going to have this kind of larger shape to make and secrete surfactant. So they're going to actually have lots of secretory vesicles in them. So they're going to be very busy trying to kind of release a bunch of surfactant. So these are going to have these kind of vesicles of surfactant. And so they're going to have to be quite a bit bigger, but we do still see that they do have the microvilli to try to deliver it more quickly. And surfactant actually kind of maintains the size of the alveoli. So it's a fluid that really reduces the collapsing of alveoli. So because of how thin they are, they could kind of fold in on themselves, and the surfactant lines it and prevents that from happening. Because I didn't do a separate thing just on cell size, the only thing I want to add here is this is our red blood cells. So I just want to make a note that white blood cells are bigger, and white blood cells are also able to grow during antibody production. So they're a little bit different from red blood cells, so they do have a nucleus, all of that. So we're just making a note that those points about red blood cells are very specific to red blood cells. Okay, next thing, so that's category 1. I'm just erasing as I go. Category 2 is specifically to look at specialized muscle tissue, And there is kind of a both but compare built into this. So I'm actually going to do it as a little bit of a table form of a Venn diagram and kind of mention what's common between them. There's actually 3 types of muscle tissue because there's also smooth muscle, but it is not listed in this category. So we are going to focus just on the 2 that it has listed. So this is skeletal muscle cells. Oh, sorry, not skeletal, just general muscle cells. Okay, and so again, I'm going to do individual things and then kind of together. So they actually, it's kind of confusing because they actually call it striated muscle, but they actually both have striations. So I usually, like at year 8, I would call it skeletal muscle. Both is going to be features that are shared, and then cardiac muscle is the specific muscle that's found in the heart. And again, when we get to physiology, we are going to do some specific stuff both on movement and on the heart. So I think, again, this is stuff that's going to come up later. So it's just kind of trying to establish a little bit of an understanding into these specialized tissue types that are going to be important down the road. Okay, so I'm going to draw them both, and then we'll end with making sure we note what's similar between them. So we've already talked about skeletal muscle a little bit before we did cell theory, but just a reminder that this is one really long cell. In terms of its diameter, it's relatively small in diameter, not tiny, but small, so 10 to 50 micrometers. But in terms of its length, it can be up into millimeters. So it can be up to 40 millimeters long. Because of how long they are, they have multiple nuclei. The nuclei tend to have kind of migrated to the ends like that. So multiple nuclei. Then when you look at it, they have bands. So when we get to muscle and movement, we're going to cover these bands because they're related to sarcomeres and movement. So they have a really important kind of reason for them. You don't have to know yet what the reason is, but you do know that, we want you to know that you see these kind of bands. And sorry, I don't mean to draw them like where the nuclei are. That's not particularly the case. And so those are the actual kind of contractile bands that show up as that. So we'll cover those in the both because they do actually have them. So multiple nuclei. So the other thing to note is that these divide differently. So they generally grow by merging rather than dividing. So they tend to actually have multiple cells that are made and then they actually merge together. So one kind of question mark is it even a cell? And the answer is kind of yes, it's been established because it has a lot of the organelles we associate with cells. So it meets a lot of the requirements, but it doesn't quite divide normally and it has 2 nuclei. So yes, but. So the answer is kind of like yes, we call it that, but it's not normal. Okay, then let's compare that to cardiac muscle. So cardiac muscle is going to be quite different. So they're going to be boxier and they tend to have this kind of branched shape. So they branch off like that. So quite a bit different. So I haven't really done a great job drawing to scale because I drew it very large. It's a bit shorter than that. So these are going to be shorter cells. And then they branch and they combine with another kind of cell here. And they are merged at these things called intercalated discs. This cell only has a single nuclei, so it's single nucleus. So it doesn't have the multiple nuclei. Like this, sorry. 1 nuclei. It does also have the striations. So we're gonna draw those on as well. So it does also have the visible appearance of these kind of contractile tissue. So these are shorter, and then they also have, they're kind of branched for connections. And And so when we get to the heart, we'll kind of cover that more, but they actually are going to be able to kind of propagate a message straight through. So with heart cells generally work in conjunction for like a single beat, whereas skeletal muscle kind of interacts in complex ways to create more different movements. So they tend to act more independently. So we tend to have long fibers that act on their own, whereas heart cells work in conjunction. So this is more, it's all about connecting the cells in this particular case. What you'll notice is the same in both cases is these striations. So striations is kind of what we call the appearance of these kind of shaded areas. So you're going to see kind of shaded areas. And so this is actually kind of shading. And it's due to contractile tissue. And again, there's going to be a lot more on that down the road. And so we can kind of go into that later in terms of what is that contractile tissue and why does it have shading in it. That can kind of come down the road, but we call the shading striations. So the tissue is actually the sarcomeres, and we'll get into that, but the result is you see these kind of bands called striations, so visible kind of contractile bands on the tissue. So that's what they have in common, that's what they have separately. So common, they both have the visible bands. What's unique is number of nuclei and size of the cells and whether they branch or not. Okay, last thing. Oh, there's one other thing I want to add, just because again, I'm worried that I haven't covered every single cell type by itself. So we're just going to make a note. Also long cells, neurons. So neurons have a similar concept, but instead of being straight, they of course have the cell body, and then it's really just the axon that's long. So that's another example of a cell that is specialized by being long, as opposed to kind of being short, big, all those different ways of shaping themselves. Okay, category 3, and this is one where it's really tricky. The actual syllabus doesn't really make clear how much you need to know in terms of, because it can get really complex in terms of the structure of these organelles yet. So when we do reproduction, we'll kind of come back. It just says adaptations of sperm and egg, and even in the two textbooks, they've kind of interpreted that quite differently, where one goes into quite a bit more detail, the other one a bit less. So I'm going to hit a middle ground of a couple of kind of specific adaptations without going way into kind of detail. Because until we've actually covered kind of how we make gametes and reproduction, it's probably a little bit unnecessary to go too far into the detail. So you definitely need to know the difference in the size. And then, like I said, I'm going to kind of include like 3 other adaptations of each. So if you did have to write on like what are the adaptations of the cells, you would be prepared to do that. But I'm not going to go into all the details. Okay, so egg. So let's draw our egg. So our egg is going to be a really large cell. So it's going to be 110 micrometers in diameter, and it is the largest cell in humans, in diameter anyway. So it's a nice round cell. Part of the reason for that is because it actually stores nutrients in the cytoplasm for the baby. So it has stored nutrients, which means it's helpful for it to be nice and big. Okay, within it has this kind of extra layer that's actually inside the plasma membrane called the zona pellucida. And so the zona pellucida has some particular adaptations to like prevents polyspermy. So the zona pellucida has receptors for sperm, but they also quickly change, and then it hardens. And the reason for that, we'll get to that kind of later, is it's an adaptation to prevent polyspermy. So it means that once fertilization happens, So we actually don't want multiple sperm to enter. So we have this kind of hardening that prevents multiple sperm from entering. So it's actually a two-step process. They have receptors. As soon as the receptor is bound, the others all shut. So only one sperm can keep working its way through. And then once the sperm is kind of in, it actually hardens the whole layer. So a couple of interesting kind of adaptations there. The way it hardens is thanks to these things called cortical granules. So they release enzymes that cause that hardening. So again, when you revise this down the road, we will have done enough on fertilization and so forth that it'll make more sense. So I think for right now, we just really want to know that it's big, that it has this extra layer around it called the zona pellucida, which has a really important role in allowing sperm in, but not allowing all the sperm in. The other thing they have that surrounds them is these follicle cells, which are essentially going to kind of moisten and protect the egg. So it has kind of this extra layer. So instead of having like a cell wall, it has these follicle cells that release fluid and kind of maintain its moisture. I'm not even going to write a specific purpose of them. We're going to get into that down the road, but just to show that they have that. So if you had to write on adaptations, you'd want to be able to write on its size. And then you can, why is it big? Because it stores the nutrients. And then you can also talk about how it has this protective kind of layer around it that is receptive to sperm, but also very quickly able to adapt and prevent other sperm from entering. Okay, so then where does that put our sperm? So here's the head of the sperm, here's the mid piece of the sperm, and here's the flagella of the sperm. So that's the flagella. It's going to mean the whole thing can be up to 50 micrometers long. In the mid piece is where we're going to have lots of mitochondria, because although the sperm doesn't have to do any kind of managing of the infant, it does actually have to swim. So it's going to make ATP for its journey. Inside the head, we're going to have our nucleus. Flagella to swim, mitochondria, ATP to swim. And then the last kind of specific thing I'm going to put in here, I'm kind of using the same color on purpose, is what's called the acrosome. And that is actually going to help break down the zona pellucida to let it in. So it's going to play a role in binding and penetrating the zona pellucida. And how it does that is it has enzymes that kind of can break it down. So again, the zona pellucida is going to kind of block anyone else from entering. Then that sperm, once it's received, it's going to kind of work its way through dissolving or breaking down enough of the zona pellucida to let it in. And then once it's in, the zona pellucida becomes like a hard wall. So there's no one else in. So we'll talk more about that when we get to reproduction. But right now, I want you to be able to kind of list a couple of features, which is to say it has a flagella for swimming, mitochondria to produce the ATP it needs for that, and an acrosome to help it actually get into the egg cell. Large cell, storage of nutrients, has a layer around it called the zona pellucida, which manages sperm entry but prevents multiple sperm entries. And then the cortical granules and follicles you don't necessarily need to get into right now, but it is worth kind of noting the cortical granules play a role in kind of creating that hardening, and the follicles in protection. Okay, that's all things B2. It's a bit longer than I anticipated, sorry, but it has a lot of weird specific things that are going to be important down the road. So worth knowing these cells quite well and these organelles so that as we build on that stuff, we already understand the structural parts of it. All right, bye. Okay, so we're on to B2.2, and this one is heavily HL, so a little bit of SL, which is literally going to go to about here of the page, and then the rest is HL. I don't think it's overly hard. It's a weird topic again because it makes a little bit of reference to things related to kind of photosynthesis and cell respiration. That you don't have the kind of functional context of yet. So again, this is more structural, so it's more understanding why they have the form they have and how that leaks to the function, but not yet the process. So a little bit all over the place, but we'll navigate that as we go. So organelles and compartmentalization. So just a couple of dot pointing things to begin with in terms of like organelles themselves. So first of all, when we say organelles, the first question we have is kind of what are they? And again, the IB has a specific definition, so rather than just like a subpart of a cell, you do have to kind of define it in a couple of ways. So it needs to be a discrete component of the cell, so it's a discrete subunit of the cell that carries out a specific function. And it can be a couple of functions, but it has to be somewhat kind of specific. So just a reminder, what does not count as an organelle? So we cannot count the cell wall because it is not within the cell. We actually do not count the cytoplasm because it's neither discrete nor does it carry out kind of a specific function. It has too many things going on in it. And we don't count the cytoskeleton because again, it's not discrete. It's kind of spread out all throughout the cell. Okay, so then the next thing is why do we have them? So that's what they are. Then the next question is why. So why are organelles valuable? So they are valuable because of compartmentalization. So they create compartments. So I'm just tired of writing the word compartmentalization, but it's linked to that. So they create kind of these distinct compartments. And when you create a distinct compartment, some of the benefits of that is that you can have high concentrations of the enzymes needed for its function. So with needed enzymes, with an ideal pH, an ease of movement, and kind of security. So those are some of the things, and we'll talk a little bit about that with the example of the lysosome. But essentially by creating a separate area, you're like, here's all the things you need for your function, and you're able to perform at your best and also kind of be separate from any threats. So it just basically is giving everything that's needed there. So in class, I kind of gave the example of setting up a science lab and having all the equipment that we need for science in one place versus spreading it all over the whole school, in which case it would be really hard to kind of get organized when we wanted to do a prac. So kind of carrying it all in one place. Last little bit, and this is just one of their little NOS things, which is like nature of science. And so you don't need to know it in any great detail. But the question becomes, how do we know all of the information about organelles? So how do we know what they do? So, I mean, of course, we can look at them in an electron micrograph and see what they look like, but where does the functional information comes from? And so we are able to study the function of individual organelles by separating out the organelles away from the rest of the actual cell and then being able to kind of manipulate that. And that's done by a process called fractionation. So fractionation, so that's essentially where we get the cells by themselves. So that's going to be like getting individual organelles by themselves. And how we do that is by ultracentrifugation. So the idea that the different organelles are different in size and therefore weight. And so because of that, we can use a centrifuge to separate, and then we kind of pour off the top and then leave these ones. And then we can recentrifuge the top lighter part and then sink those. So this kind of stepwise centrifugation, which gives us isolated organelles. And then once we have an isolated organelle, then we are able to actually study its function. So that's our little bit of just kind of information generally. What I want to do with the rest of this section is to create another table. It's actually fairly similar to the last table of going through the organelles. So what are the organelles that you need to know? And then do a diagram that covers what you need to know about them structurally, and then have a little bit of an explanation of how its structure links to its function. So there's only two that are going to be, but I'm going to go ahead and erase this anyway, just so I can have room for my whole diagram. So the first two you need to know about for SL. So you do need to know about kind of why is the nucleus compartmentalized away from the cytoplasm. And then you need to know the lysosomes as an example of kind of cytoplasmic compartmentalization. And then for HL, you need to pick up mitochondria, chloroplast, ribosome, Golgi, and then a little bit of a random sub thing on vesicle formation. Okay, so let's do organelle. Boom. Drawing, which is basically to show its structure. And then we're going to say link to function. So in other words, how is what we've drawn contributing to this function that it carries out? Okay, so first up, the nucleus. So we'll start with that. So in terms of what you have to know about the nucleus is a little bit about its double membrane structure, the fact that it has pores and the fact that chromatin inside of it kind of keeps it expanded. So we're going to draw a little double membrane, which is just so much fun. So there's kind of one phospholipid bilayer, and then there's another phospholipid bilayer. And then running through that, we use protein channels to create a pore. So the pore needs to be a bit bigger than a normal protein channel. So it's going to create a decent size kind of opening. And that's going to go through both layers of the membrane. So just trying to show the double membrane and then also trying to draw a pore. I'm not going to draw any more double membrane because it's no fun. So I'm just going to draw it kind of like this. Okay, so there's my double membrane. Which is important. Here's my pore. And then the third thing that we want to understand is where does the actual DNA fit in? So chromatin is DNA that's already wrapped around the histones, and that's going to basically come over here and interact. Sorry, I haven't drawn it wrapped very well, but it's going to basically kind of hold on to the inner membrane and kind of bind to the inner membrane. And the reason it does that is to essentially hold the shape. So it kind of stretches out the chromatin and kind of holds against the different parts of the inner membrane. So it actually acts as like a cytoskeleton within the nucleus. Okay, so then let's explain kind of our links to function. So first thing we want to clarify is that it's really important that it is separate from the cytoplasm. And the reason for that is it ensures that all the DNA processes are happening in their own location. And this is going to be way more clear once we get into D1. But what you need to know is that it means transcription happens here before translation, which actually means there's a pause between transcription and translation, which allows some modifications to be made. So for right now, we're just going to say that it separates transcription and translation. And that actually allows for some mRNA editing or processing. So we have the ability, because it doesn't start automatically being used right as it's made, it actually allows time to kind of make some changes to the mRNA, to add some protection for it, remove some introns, things like that, remove some exons, remove the exons from the introns. And so that's all going to be really important down the road. So right now we just want to know that that's actually really valuable to have the processes separate from one another. Next thing we want to know about is the double membrane. And the double membrane is nice because it easily forms vesicles. So it forms vesicles to scatter during cell division. and then they merge together again kind of at the end. So by having the double membrane structure, it actually pretty easily kind of self-vesicles. So it can form a pretty easy vesicle formation and then move away and kind of dissolve, but it hasn't actually dissolved, it's just packaged away. And then when it's time to kind of recreate the nuclear membrane at the end of cell division, the vesicles come together. So that's a real benefit. That is actually an HL only point. So anyway, just if that confuses your SL, you don't really have to know that. But we do want to know that it has a pore for mRNA and ribosome movement. And it does other things. I'm going to say etc. It also allows transcription factors in. It allows ribosomal subunits out. So there's a few different reasons for the pore, but we do have kind of an ability to kind of transfer things between those compartments. Okay, so that's the things you need to know about the nucleus. Next up, we're going to have the lysosomes. And so it's interesting because it's actually labeled as compartmentalization of the cytoplasm, with an example being the lysosomes, and that's the only example you need to know at an SL level. HL, you're going to know more. So I'm just going to put a note that this is given as an example of cytoplasm compartmentalization. And so it basically is showing us some of the values of compartmentalization in this particular example. So here I have my cell, and my cell has actually just absorbed a pathogen. So I have a pathogen like that. And so now that pathogen is inside the cell, and it's inside its own little vesicle. So that is an example of endocytosis. It's actually called phagocytosis, because it's a particle, or you can call it like pathogenesis. It has different terms. But anyway, it's an example of endocytosis. And then over here, I have my lysosome. And lysosomes are really interesting because they have all these hydrolytic enzymes in them. And hydrolytic enzymes break things down. And so if they were actually just open in the cytoplasm, then what would end up happening is that we would end up damaging the cytoplasm. So this is actually going to come over here and merge with that and actually release its actual enzymes directly into the actual pocket that's holding the pathogen. So it's an example where this is a membrane that keeps it safe, and then it also can fuse and deliver its enzymes directly to what I need to break down. So that's a really good example, is that it kind of increases the concentration within the organelle. While preventing it from being in the cytoplasm. And that's done by having a vesicle. So again, it's increased concentration inside of it, prevent it from damaging others because of the vesicle membrane. And also, just to clarify, the lysosome is an example of a vesicle. So it is a vesicle of hydrolytic enzymes. And then it also can merge and release the enzymes directly where they're needed. So it's able to kind of act exactly where it's needed as a result. And then again, by keeping it separate, we're not damaging other enzymes. So that's just an example where compartmentalization does some of the things we said before. It has a nice concentration of enzymes in one place, and it actually is able to move more easily. So it can kind of move and direct itself towards what it needs to break down, and it provides a safety mechanism by being inside of its own membrane. Okay, so those are the only two kind of examples that you need to know at an SL level. At an HL level, we have four more. Okay, so the next two for HL is going to be the chloroplasts and mitochondria. And these are going to be a really big component down the road when we do photosynthesis and respiration properly. So you need to know why the parts of them facilitate it, even though we don't know the process. So it's a little bit confusing, so we're just going to do our best for right now to kind of get the information you need for here and know that it'll make more sense down the road. So we'll start with the mitochondria. And honestly, you already kind of know a lot of this anyway, because in A2, you had to go into a little bit of its structure to recognize it. So it should be relatively familiar, the names of the different places, but we just need to talk a little bit more about how they look and that they look that way for a reason. Okay, so here is my mitochondria. That's my outer membrane. Then my inner membrane looks something like this. Sorry, I'm trying to not have it absolutely run into the outer one, so there does need to be a small space. Like that. Okay, so that's going to be my inner membrane. So I can add the word membrane. And then the little spaces that it creates, that's called the cristae. So the cristae is actually kind of the gap between because of the folds. So it folds in and creates these little like indentations called the cristae. So that's the cristae, is essentially the folded in part. And then the area between the two membranes is the intermembrane space, including the cristae. So both of those. So there's a little bit of space just down at the bottom as well, but the cristae are intermembrane space. So that's kind of the intermembrane space, or we can call it as the cristae when we're specifically meaning the area created by the folds. Okay, so that's what we need to know. I'm not redrawing I will label the matrix. I'm not drawing the DNA and all of that. That's really more a different topic. And then the inside part is the matrix. Okay, so let's look at some of the functional purposes here. So there's kind of three things I really think you need to know, which is why is there so much membrane? Why is the intermembrane space small? And what is the benefit, what's going on in the matrix? So that one should be pretty straightforward. So first of all, why do we have so much intermembrane? So it has the folding, and the folding does two things. So it increases the amount of intermembrane, which another way that you could say that is that it increases the surface area, or the surface area to volume ratio, which we'll cover in our next one. But now we need to say, okay, well, why is that important? Like, what's going on in the inner membrane that's so important? Well, my inner membrane actually contains the electron transport chain, and ATP synthase. So because it contains the things that make most of the ATP, so if we say more membrane, so more inner membrane equals more ATP, right? So if the inner membrane is where we're making our inner ATP through, then we want to have lots of it. The next thing is, why is our intermembrane space small? And this is the part that's confusing till we've actually gone into respiration. But essentially, ATP synthase, so kind of like we talked about in the last part about how we can move glucose if we build up a sodium concentration. So it works a similar way. So we can get ATP synthase active if it starts moving hydrogens. And so we need to build up a hydrogen proton gradient in the intermembrane space. So the flatter that is, the quicker we can build things up. So I used the example in class, if I gave you a really small amount of water versus a big glass, and I gave you drops of food dye, and I was like, how quickly can you get this to be blue? It's gonna be a lot easier if you have this small amount of water. It'll very quickly turn blue, whereas if you have the larger, you would have to add more. So we want to have a thin intermembrane space. And the reason for that is it builds up a hydrogen proton gradient faster. And then we want to make our link. Why does that matter? Because a hydrogen proton gradient is needed for ATP synthase. And so when you are revising this after we've done the rest of C1, like, oh yeah, right now you just kind of have to learn why. So just almost memorize it for right now, and then we'll come to like where that all fits in later. And then the last thing is that the matrix is where the Krebs cycle happens, which involves a lot of kind of intermediates and enzymes. So the matrix has a high concentration of the enzymes and substrates needed for a Krebs cycle. So Krebs cycle, when we get to it, involves a lot of kind of recyclable substrates and things like that. So it has a lot of things that we need. So by having that actually, it's a terribly unstraight line, it's just now stressing me. By having it in one kind of place, we can kind of gather all of that there and kind of concentrate it in that central area versus having it kind of spread out. Having said that, most of the mitochondria is matrix, so it's not overly concentrated. And then when we say the intermembrane space, a note that we can say for the cristae is that it makes more intermembrane space. So if we didn't have and more intermembrane. So it kind of does both of these. So the cristae does this and this because it adds those extra little thin indentations. Okay, so that is the mighty mitochondria. And then we'll go chloroplast. And again, everything from this point onwards is HL only. So I'm just writing that for completion's sakes, but that is kind of implied. Okay, so let's draw our chloroplast. Somewhat similar to a mitochondria, but the inner membrane just sits close to the outer membrane, kind of like in the nucleus, and it doesn't actually use the intermembrane space. So that is a kind of distinct difference. Then we're going to have our thylakoid stacks. They're going to be a little connected by some lamella. So, okay, so then let's label our key things. So each of these is a thylakoid. They're stacked together to form a granum. And then the area in between is the stroma. Okay, so then kind of we're going to again do 3 things to know about structure to function. So first of all, that each individual thylakoid is very thin. So thin thylakoid. So let's kind of clarify why. So inside the thylakoids, they contain ATP synthase. And so same idea. So by having them be thin, it accumulates the proton gradient quickly for the ATP synthase. And this is where it gets a bit tricky because you're like, what? ATP synthase? I thought ATP was the respiration, but you do make ATP in the first part of photosynthesis. So that's kind of important for that. And then why do we have so many stacked thylakoids? So we're stacking them into a granum, but why do we have many and why do we stack them? So similar to above, the reason why we do that is because it increases the amount of membrane, which remember we can say increases the surface area to volume ratio. And we already kind of covered ATP synthase is there, but there's an extra thing that we want to know, that in the membrane there are photosystems. And there's also ATP synthase. So the more we have, the more membrane, the more photosystems, the more ATP synthase. So all of this means kind of important for the fast light-dependent reactions. For right now, you just want to be able to make the connection that we want lots of membrane so we can absorb lots of sunlight and get that first stage of photosynthesis going. So light-dependent reactions is stage 1 of photosynthesis, and we want it to go quickly and efficiently. And then similar to the matrix, then we're going to have the enzymes and substrates needed for the Calvin cycle inside the chloroplast. Oops, in the stroma. So inside the stroma is where the Calvin cycle is going to happen. And again, because we're calling both of these cycles, we're recognizing they use a lot of enzymes. They're a big metabolic pathway. Lots of enzymes needed, lots of substrates needed. So by having that kind of localized to one area, we can increase the efficiency of part 2 of the process as well. So part 1, part 2. So that's kind of the 2 stages of photosynthesis. Krebs cycle is stage 2, and the electron transport chain is stage 3. You can piece together where those fit with the processes once we've learned them, but right now we just want to make sure we understand some of the ideas behind why there's so much membrane in them and so forth. Okay, last thing, so 2 more organelles and then vesicles, which are arguably in some cases an organelle, but a slightly kind of strange placement. We're just going to put it in our chart because it's nicer to organize it that way. So it is an actual dot point. So the dot point has you know about ribosomes. It doesn't actually have a specific dot point on endoplasmic reticulum, but we will kind of cover the rough ER in the context of ribosomes. Okay, so our next organelle that we will talk about is ribosomes. This is not overly complex, but it is nonetheless HL material. And really what they want you to recognize is that there are actually 2 different places that you see ribosomes in a cell. So we actually have free ribosomes floating around minding their business. And then we also have ribosomes that are on the rough ER. So then over here, let's draw some rough ER. And then we also have ribosomes that sit on the rough ER. So this is a free ribosome, and this is an attached ribosome. The free ribosomes are going to build proteins that are going to go out into the cell. So they're just going to go out into the cell, whereas this one is going to build a protein that's going to go into the ER. And then ultimately that ER is going to then branch out and make a little vesicle for it. So this one's just going to be free. So out here floating around is a little protein, whereas that one is going to put it in a vesicle by the rough ER. So that's really what we want to know about. So just a reminder, what are they? They are actually a single membrane. We just did 2 double membranes. And they're made up of rRNA and protein. So they're a little protein with rRNA wrapped around it. Free ribosomes make proteins for the cell. Or for organelles within the cell. So they're going to make proteins that are released directly into the cell. Attached ribosomes make proteins for export usually, or for large distances. And they get they go into the rough ER and into a vesicle. So the kind of pathway that the proteins take looks different, but they do the exact same thing, which is that they make proteins, and it's probably a fairly obvious thing that if it was floating on its own, it wouldn't have access to the ER, so it'd be more likely to just be kind of directly administered. Alrighty. Next is the Golgi. So that's going to be kind of my next stop along. So we're in my Golgi apparatus. And you need to know a few kind of specific details in terms of its structure. So that's HL. So first of all, here's going to be my ER that we just showed. And here's my protein that's coming in the vesicle off the ER. And then it's going to come over here and travel to the Golgi. And then over here we'll have the cell membrane. Okay, so the couple of terms you need to know. So each stack is called a cisternae, just a term to memorize. And then you just have to memorize which side faces which. So the side that faces the rough ER is the cis side, or cis facing, and the side that faces away is the trans side. Okay, so then this is going to ultimately come over here and merge in and deliver the protein. The protein is going to go through some modifications and become like a nice little globular protein, and then ultimately be formed into a new vesicle that is going to go out through the cell membrane. So it both receives vesicles and creates vesicles. So very vesicle oriented. So basically it modifies and packages proteins into vesicles for exocytosis. So again, here's our example of exocytosis. Is occurring when we make proteins in the cell and then export them out of the Golgi. Okay, last little thing, and again, it's kind of a slightly weird placement. Probably would have made more sense with endocytosis, but anyway, we will include it as one of our little organelles. So I'm going to include it in the same color to show that it's very linked to the Golgi normally. But in this case, we're actually looking at what's going to happen at endocytosis. So exocytosis there. Okay, so here's my cell membrane. I'm having regrets about making this pink. If you left it pink, that's fine. Because this is a vesicle of the cell membrane. Okay, and then on here we have receptors. So a lot of times endocytosis is not random, like it's not just something giant actually pushing in. One really cool thing is the cell can kind of deliberately look out to bring things in. So it can have receptors for things that it wants to bring in. So it might be that it's needing these proteins inside. And so it goes ahead and has receptors on the cell membrane to bring these in. So this is like sending invitations, like please come in. So these are inviting these things in. We want you to come and join our cell. And so here's the actual what you're wanting to bring in. So this is the needed particle. And then what happens is to hold all of the receptors in place. So if they can be really close to each other, it's actually easier to bring them in if they're really close together, because then we can bring them in one vesicle. So rather than spreading out these receptors and having to kind of bring each one individually, this actually brings them all in a big, nice little bubble. So then what happens is when that gets filled, then the membrane envelops around those vesicles. And so then this actually invaginates in like so. So that whole area forms, and these clathrins, I drew, there was more of them there, actually pull around each other to form a quick membrane. And so then all of these particles move in that way. Then the clathrins will recycle and so forth. So these are the clathrins. So they're really important because they actually enable you to quickly form those. So in terms of vesicles, if I go back to what I was saying here, the example up here these are actually, and I'm going to just add it up there. These vesicles are actually called secretory vesicles because they're leaving. If what we've drawn here is an intake vesicle, so this is actually an example, this process here is receptor-mediated endocytosis. And so these are incoming vesicles. So these are incoming, not secretory vesicles. And so this is aided by clathrins. And so these are just proteins that anchor receptors. And they actually allow for fast vesicle formation. So they provide the anchoring, and then they also kind of pull in quickly, so that allows for endocytosis to happen quickly and also allows for it to be kind of specialized and looking for things that are needed in the cell. All right, so that's a wrap on B2.2. Last we have B2.3, is probably somewhere in between the last two, so a little bit of complexity in terms of specialized cells, but not quite as intense. It's again going to be fairly short on the SL, so SL will be short, HL will be longer.